So today you're going to take Cornell notes on the cell membrane. Remember that your notes need to have your questions on the left and at least a 5-sentence summary at the very end. Now, cell membranes are so extremely important. And an analogy that we can use to kind of describe the function of cell membranes is the offensive line in football. The offensive line wants to let the running back with the football through the line, but it doesn't want to let the defensive line in. So cells want to allow specific things to be able to go in and out of them, but they need to protect themselves from other harmful things. So first, let's look at the structure of the cell membrane. I do want you to draw the structure, which you see up on the right-hand side. The structure of the cell membrane is it's made up of a lipid bilayer. And remember, lipids are one of the macromolecules that we learned about at the very beginning of the year. Here you can see that they have a polar head, which is the circle, and it's hydrophilic, and it has two tails. And the membrane lines up where the tails are kind of facing towards each other and the heads are on the outside. Now, this lipid bilayer is made up of what are called phospholipids. And the phospholipids have the polar head, which has a partial positive charge on one side, or pole, and a partial negative side on the other. And that makes them hydrophilic. And what hydrophilic means is that they love water. The tails are nonpolar, so there's no positive or negative poles or ends on the tails, and that makes them hydrophobic. And hydrophobic means that they hate water. So because of this feature that the heads are hydrophilic, water loving, and the tails are hydrophobic, water hating, they automatically form this bilayer where the tails are on the inside because they don't want to touch any water. And the heads do like water, and so they automatically form into this layer.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
All right, there are 3 types of proteins that are in the cell membrane. The first are channel proteins, and channel proteins are passageways that allow specific things to enter in or out of the cell. In the animation that you see below, you'll see 2 different channel proteins. One that allows the sodium ions to go into the cell, and one that allows the potassium ions to exit out of the cell. Receptor proteins are the second type of membrane protein. What they do is they transmit information into and out of the cell. How they work is that an external molecule from outside the cell will lock onto the receptor protein, and it will cause a reaction that takes place inside of the cell, and it will allow the cell membrane to do specific tasks or send specific messages. Next is the marker protein, and marker proteins are name tags that identify the type of the cell and who it belongs to. This is so extremely important for our bodies, especially our with our immune system. So for example, if you have a bacteria that enters into your body, your immune system doesn't recognize the cells. It doesn't recognize its marker proteins or its name tag, and so it's going to attack and destroy those cells, the bacteria. This can also play a role if someone is getting a transplant. For example, let's say someone needs a kidney transplant. When they get the new kidney, it has marker proteins from the original owner of that kidney. And so your immune system, when you have that new kidney implanted in you, you will your body will not recognize it because it doesn't have the right marker proteins. And your immune system will form an attack against that kidney and reject the transplant. And so that's why transplant patients are put on specific immunosuppressants, which is going to suppress their immune system so that their immune system doesn't attack that new kidney that they get. Now, these marker proteins are made up of chains of sugars or carbohydrates that are attached to the proteins in the cell membrane, and they basically make a flag that identifies the cell. And so here you can see these blue things. Those are the chains of the sugars that are attached to the protein. And again, they are marking the cell or naming the cell, identifying it. So there are some analogies that you could use to help you remember what the different proteins do. So a channel protein, kind of think of them as a bouncer at a club. They are only going to allow people with the right identification into the club, and only if there's enough space available in the club. And they're going to kick certain things out of the cell as well. Now, a receptor protein is kind of like the intercom at an apartment. So at the door, there's the little intercom where you press the button for the person that you want to contact. And if that person says, okay, you're the right person, you can come on up, they're going to press a button which will unlock the door and allow you into the apartment. If you are not someone they know, they're not going to let you in. So that's kind of what the receptor protein does. Something comes from the outside, and if it's the right one, it will have a process that will occur inside of the cell. It will send some information. And lastly, the marker protein is like a name tag, or it could be like a flag that identifies what the cell is. Now, a cell membrane can be described in two ways. First is it's a fluid mosaic. And what that means is that the phospholipids and the proteins that are in the cell membrane, they're not fixed or stuck in place. They can move and wiggle around and kind of change their locations. Next is semipermeable. So semipermeable is where only some molecules are allowed to go in or out of the cell. And so here, this red dotted line is the cell membrane, and it's only allowing the blue and the white ones to go in and out. The green ones can't fit, so they can't go through that cell membrane. So that is semipermeable.